In this video, we will continue our discussion of viral replication. Our focus will be on the replication of animal viruses and latency of animal viruses. Animal viruses follow the same basic replication stages as the bacteriophages. However, animal viruses demonstrate some notable differences from the bacteriophages in the exact details of each stage. Animal viruses differ from bacteriophages in the manner in which they enter the host cell, due to the presence of an envelope on animal viruses. The synthesis of viral components is different due to structural differences of the prokaryotic host cell and the eukaryotic host cell. Finally, the manner in which animal viruses are released is different due to the absence of a cell wall on animal cells. Animal viruses are structurally different from bacteriophages in that they lack tails and tail fibers. Therefore, attachment requires an exact fit between proteins of the capsid or glycoproteins of the envelope of a virion with a receptor on the cell membrane of a host cell. There are three mechanisms utilized by animal viruses to gain entrance to the host cell following attachment: direct penetration, membrane fusion, and endocytosis. The process of direct penetration occurs when the capsid of a naked virion sinks into the cell membrane, creating a pore through which the virions inject the nucleic acid into the host cell. Envelope viruses may gain entrance into the host cell through membrane fusion. The glycoproteins of the viral envelope attach to receptors on the host cell membrane, and then the envelope and cell membrane fuse, releasing the nucleocapsid into the cytoplasm of the cell. The viral envelope remains as part of the cell membrane. Both enveloped and naked viruses can enter by endocytosis. The virus attaches to host cell membranes utilizing either capsid proteins or glycoprotein spikes. The host cell membrane then invaginates to form a vesicle containing the virus. Membrane fusion and endocytosis both require an additional step, known as uncoating, in which the capsid is removed to free the viral nucleic acid. The mechanisms by which the virus undergoes uncoating varies greatly among viruses. Some are uncoated within the vesicle, while others undergo uncoating in the cytoplasm as a result of enzymatic action. The free viral nucleic acid will direct the cell to begin synthesizing viral components. The events that take place are dependent on the kind of nucleic acid. The nucleic acid of the DNA virus generally enters the host nucleus where it is replicated. RNA viruses, on the other hand, are replicated in the cytoplasm. Regardless of the type of viral nucleic acid, it must accomplish two tasks. It must synthesize more viral nucleic acids and more viral proteins. In order to synthesize viral proteins, the virus must first synthesize a mRNA molecule to be read by the host cell's ribosomes. We will examine the process by which each type of animal virus synthesizes new viral components. The nucleic acid of double-stranded DNA animal viruses are typically replicated within the nucleus of the host cell. The host cell's DNA polymerases are often used to help synthesize the new viral DNA. The process by which the nucleic acid is replicated is similar to what takes place during replication of the host cell's DNA. Each strand of the original nucleic acid serves as a strand to synthesize the complementary strand. In addition to synthesizing many copies of viral DNA, the host cell must also synthesize viral proteins. The host cell transcribes the genes into mRNA, and the host cell's ribosomes translate the mRNA into viral proteins required for the capsid and other structures. Single-stranded DNA viruses are also replicated within the nucleus of the host. The single DNA strand is utilized to synthesize the complementary strand of DNA. The two strands then form a double-stranded molecule, which is utilized to carry out the same processes seen in the synthesis of double-stranded DNA. RNA viruses are typically synthesized within the cytoplasm of the host cell. RNA viruses exist in several forms: positive sense single-stranded RNA, negative sense single-stranded RNA, retroviruses, and double-stranded RNA viruses. Positive sense RNA molecules are RNA that can easily be translated by the host cell's ribosomes. The negative sense RNA molecules must be converted to its complementary strand, which is a positive sense RNA molecule, before being translated by host cell's ribosomes. Positive sense single-stranded RNA viruses must first translate a gene from its RNA that codes for a viral RNA polymerase. The viral RNA polymerase is then used to synthesize a complementary negative strand RNA to serve as a template to make more positive strand RNA. The viral RNA polymerase must be synthesized because animal cells do not synthesize RNA molecules from an RNA template. Translation of other genes on the positive strand genome produces the necessary proteins for capsids and other structures. The retrovirus has a positive strand genome that must be converted to a DNA genome. The virus carries with it an enzyme known as reverse transcriptase. Reverse transcriptase synthesizes a strand of DNA complementary to the RNA genome and then directs the synthesis of the complementary DNA strand to yield a double-stranded viral DNA molecule. The double-stranded viral DNA moves into the nucleus where it is integrated into the host genome and is transcribed to yield more viral RNA. Translation of some of the viral RNA yields viral proteins for virus assembly. The human immunodeficiency virus is an example of a retrovirus. Negative sense single-stranded RNA viruses carry with them an enzyme known as RNA-dependent RNA transcriptase. The enzyme transcribes the negative strand RNA molecule to complementary positive strand RNA molecules. The positive strand RNA is then used to make many copies of negative strand RNA and used for translation at host ribosomes to create viral proteins. Double-stranded RNA viruses contain both positive and negative strand RNA. The positive strand molecule is translated by host cell ribosomes. A viral RNA polymerase is produced from the positive strand early in the synthesis process. The viral RNA polymerase utilizes the positive strand as a template to synthesize more negative strands, and the negative strand is used as a template to synthesize more positive strands. This process is similar to replication of a double-stranded DNA molecule. Later in the process of synthesis, the positive RNA strand is translated to produce other viral proteins. As the pool of viral components accumulate within the cell, they begin to spontaneously assemble to form new virus particles. DNA viruses typically assemble within the host cell's nucleus, while RNA viruses generally assemble within the cytoplasm. The number of viruses formed and released varies between viruses and is dependent upon the health of the host cell and the type and size of the virus. They may release anywhere from a few thousand virions to over 100,000 virions from a single infected cell. The mechanism by which a virus is released from the cell is dependent upon the presence or absence of a viral envelope. Those lacking an envelope are referred to as naked viruses. Naked viruses may be released through exocytosis or by lysing the host cell. When naked viruses exit the cell through exocytosis, they do not acquire part of the host cell membrane. Envelope viruses are released by a process known as budding. Envelope viruses must insert viral glycoproteins into the host cell membrane before exiting the cell. Envelope viruses may acquire their envelope from the nuclear membrane, membranes of the endoplasmic reticulum, or cell membrane. Envelope viruses do not readily kill their hosts, so the cell may remain infected, slowly producing and shedding viruses. Such an infection is known as a persistent infection. Some host cells are not immediately harmed by a virus, but rather harbor the virus. Cells can harbor viruses for a few weeks, and in some cases, the virus is harbored for the remainder of the individual's life. Some of these viruses are capable of becoming active periodically. Such viruses are referred to as latent viruses or proviruses. Examples of such viruses are the herpes viruses. Animal virus latency differs from lysogeny in bacteriophages because animal virus nucleic acids may or may not become incorporated into the host chromosome. If the viral nucleic acid is integrated into the host DNA, it will not be excised because induction does not occur in eukaryotic cells.